Hi, everybody. My name is Vince. I'm with Top Doctor, and we're really excited today to introduce Dr. Mukesh Kumar. He is probably, if not the, definitely in the top 3 FDA consultants in the world. So we actually have one of the top FDA consultants in the world. Dr. Kumar worked for NIH as an employee for around 7 years, and then he contracted with the FDA, or he was a contractor with the FDA for over 15 years. So right now, he is a consultant to many companies. And let me just give you a little quick background. Dr. Kumar, his first name is Mukesh, Dr. Kumar Mukesh, in the last 3 to 4 years, has represented, consulted for, helped, basically, around 14 companies sell for more than $1 billion. This is the pharma world, obviously, and also he is an expert in every facet of testing. More importantly, we're here to talk today about the COVID-19 rapid antibody test, and we believe that this Reszon diagnostics test is the best test on the market. And there is some evidence to support a bit of this, and I'm going to turn it over to Dr. Kumar right now.
Thank you, Vince. It's my pleasure to talk about Reszon Diagnostics' latest product. It's a product that detects antibodies to COVID-19 virus. As you know, when virus infects your body, you make antibodies, and by looking for the presence or absence of antibodies, you can measure if somebody have an active infection or was infected and has recovered. These tests are very important to manage the treatment of COVID-19. As you would notice, one of the key aspects of these tests is the ease of how these tests are done. These tests are done with a drop of blood. They look like a cassette, something similar to as you see in a pregnancy test, where you drop a drop of blood and then a buffer, and you see something happening on the slide. You see colors appearing, lines appearing, and that tells you if somebody has antibodies to the COVID-19 virus. These kind of tests are very common. This is not the only test of its kind. This technology has been used for many diagnostics. The technology is called lateral flow. Lateral flow test is something like think of it as a microscope slide where you have antibodies and receptors to them immobilized, and you add your analyte on one end, and the analyte flows through the slide. Basically, a slide is made of a paper, absorbent paper, that allows the reagents to flow across the slide and bind to different things. So in our case, we have 2 antibodies, primary and secondary antibodies, and the analyte over here is a drop of your blood. It could be a drop of your serum. It could be a drop of your plasma. That becomes very important, particularly if somebody has a stored sample of blood. So you add your analyte, and then your analyte flows through the slide. And when it binds to the location where you have the primary antibodies, it creates a color response. You see a colored line appear at that place. This is before the reaction. There is no line, and when the reaction happens, you see a line, and you can have controls right built in the slide, so you can see if the test is working or not working properly. So you have controls right there, and the entire process takes minutes. You can see your results as early as 5 to 7 minutes. You can read them for as long as 20 minutes. It's not recommended to keep them for too long, because if you keep them too long, you start seeing too much background, and the test is no longer reliable. Reszon test, is developed.
Hang on a second,
Sure.
Dr. Kumar.
Yeah.
But the best results are after 15 minutes and maybe before 20 minutes, because I'm reading the instructions. And
yeah,
after 30
so
minutes,
typically,
throw it out.
Yeah, typically the process, the liquid has to flow all the way back, all the way across all the way across to the, if you can see this slide, the liquid has to go all the way to the end of the slide, and it takes about 10 to 15 minutes. 10 minutes is the minimum time you would see. Generally, in about 15 minutes, you'll see it completed, the flow has completed, and that's when we recommend for people to get the best result, because that means the entire amount of antibody in your analyte has flown through the slide, so you get the best response. For viral infection, like any viruses, antibodies appear at different times. So if you see the blue arrow, the blue line represents when somebody gets infected by the virus. So if you get in contact with somebody who was infected with the virus, you in the beginning, for some time you will have no symptoms. That's called the asymptomatic stage, and then you start seeing antibodies. Antibodies appear around the same time when you start showing symptoms of the disease. The first antibody, which is the yellow line that you see, is the IgA antibody. It's an antibody that's present in your saliva and mucus and other places. Then the green line is your IgM antibodies, and then you have the IgG, the red line. The IgG antibodies are the ones that are also called the memory antibodies. These are antibodies that will stay in your body all the time, and if you ever have a second infection by the virus, these antibodies will fight that infection. So this is how usually infection works. Now, as you can imagine, when you are looking for antibodies, the best time you start seeing antibodies is somewhere around day 7 or 14 of infection. So generally, at the peak of your infection, you start seeing IgM antibodies. As you can see, the peak of IgM coincides with the peak of your infection, the blue line. And then as the infection goes down, the IgM level also goes down and IgG level starts rising. Now, the reason it's so important is because by detecting both the antibodies, IgM and IgG, you can make a good guess about the current infection stage of a patient. Now, obviously, antibodies are not the virus. You are measuring the antibodies. So you cannot say with surety if somebody is infected or not, but it's a good indicator of infection. And not only that, by looking at IgM versus IgG, you can distinguish if somebody has an early infection or somebody has a late infection or somebody has recovered from the infection.
Can I ask you a question while you're here, proposed to us by doctors? So they'll get an IgM, okay, and that means they're in a viral stage. A virus has started, correct?
That means they're highly infected.
Well, if they're asking, what do they do? So do they send them on for another test or?
Yeah. So the recommendation is from CDC and FDA is that if you do see antibodies to the virus, irrespective of IgM or IgG, if you do see antibodies in your serum, the recommendation is to do an antigen test, which is you look for the virus, and those are your PCR tests, where you look for the viral DNA or viral RNA in the patient sample. So that is the CDC recommendation, because antibodies are not by themselves a definitive diagnosis of infection. They are very quick, so they are very useful for screening. If you're screening a large number of patients, if you have hundreds of people lined up, the PCR test takes about a week to get the results. The fastest PCR takes about 3 to 4 days turnaround time. As you can imagine, if you are doing a large population, you don't have that kind of time. So antibody tests, which can be used to screen a large number of people in a very small time and see who needs to be tested for PCR. And that's what CDC and FDA recommend.
And if you just have the IgG, let's say you have the IgG antibody, you should not currently have the virus, correct?
Most likely. Again, most likely you could have small, as you can see, there is some overlap in the blue line. Although the virus is going down, there is some virus while you have IgG. So presence of IgG does not mean that there is no virus at all. It means the virus is probably, the infection is on a decline, but it's not confirmatory. You cannot say for surety that because you have IgG, you're not going to have virus. Because you can see, if you see on the right-hand side of the curve, there is overlap. There's overlap between the virus, the blue line, the green line, the red line, and the yellow line. So there are times when you have the virus and also the antibodies, and there is time. If you see, the red line goes way beyond when the virus is gone. So you do see antibodies for a very long time. Actually, that is the antibody which FDA call as convalescence plasma or blood, where you now can donate your blood, so somebody can isolate IgGs from your blood and use to treat other people. So you do want to know if you have IgGs or not for the virus, but you still have to get a second test. CDC recommends any IgG test, any antibody test be confirmed by a second test, such as a PCR test.
And by the way, for people that aren't familiar with a lot of lab testing, in almost every phase of lab testing, from toxicology to pregnancy or metals, there is almost always a confirmation test. They want a confirmation.
Yes.
You test
Yes.
and retest. So this is not atypical of the COVID-19 tests on the market.
That is correct. Almost every test that you do, even simple tests, need to be confirmed by second. It's similar to when you see symptoms of a flu or symptoms of HIV infection, you confirm them with multiple tests. You don't do just one test. Or cancer or any other disease, infectious disease or non-infectious disease, you always confirm with multiple tests to be sure before you start treating people. Antibodies are used universally for all kind of diagnostics, not just COVID-19. These antibody tests are very common, and there are antibody tests for everything, every infectious disease you know of, and all of them require confirmation by a second test. So moving on, the beauty of these antibody tests, as you can see, is very easy. They're very easy to interpret. In the Reszon test, for example, these are just pictures of different outcomes that you can have with the test. You can have a negative, somebody who has no antibody, in which case you will only see the control line. And then you have the G line or the M line. The presence of G line would indicate you have IgGs. The presence of M line would indicate IgM, which typically happens when you are early in the infection. And you could have both IgG and IgM lines, as you can see on the top right-hand panel, which, as you saw in the previous slide, there is overlap when all IgG and IgM both could be present. And the control line is very critical, is very, very useful. If you do not see the control line, that means the test is invalid, as you can see in the bottom panel. So it's very easy to know if you did it right. I mean, typically when you do a test, you are always worried, did I do the test right? I mean, if I'm seeing negative, is it truly a negative, or if I did something wrong? In these tests, it's very easy to know that. If you do not see the control line, that means something did not happen properly. Either you did not do it properly, or the slide was defective. In either case, you repeat the test. And
so in every case, you need to, for it to be a valid test, you need to see the C line, the line under the C.
Yes. The C line always has to happen, and actually that is the best part of this kind of test, because the control is right on the slide. So it's not that you have to you have to run a second slide, or you have to run something else independently to see if it's happening or not. You see it right on the slide if the test happened properly or not. So that, and the Reszon test, I mean, it's pretty simple to do. It takes practically very little training. It comes with detailed instructions what to do. It comes with all the things you need. It comes with it comes with a pipette, with a little dropper to collect your blood. It comes with a little lancet to prick your finger. It also comes with the buffer and alcohol wipes and everything. So it's fully ready to use. You don't need anything else to do this test. And this test has been validated robustly. It has been tested in 3 different countries in about 200 plus patients. This was a clinical trial. It has been used in more than 3 million patients in the 3 countries, between France, Malaysia, and China. It has been tested in more than 3 million patients since April, since the last 3 months. And the experiments that were done to validate these tests were done in 200 plus patients, in which it was found that its sensitivity is about 98% for IgGs and for about 97% overall. In terms of negative tests, giving it, a false negative, it is about 100%. It was found about 100% accurate in terms of giving you only positive tests and not giving you negative tests. It was tested against all kinds of other things that can interfere with the test. The testing was done in compliance with all international testing requirements, and the test is still being, there are additional tests being done to validate it even more. But the test has been marketed since April, and it has been, it has been found to be very accurate and sensitive.
So really impressive is the French data, because they tested a bunch of other tests, correct? At the same time?
Yes. Actually, in all three cases, French, Malaysian, and Chinese tests, the test was compared to other tests. There were other tests available. The two tests that were used were both manufactured in China, and the test was compared to them, and it was found to be much more sensitive and much more accurate in terms of the relative sensitivity to detect IgGs and IgM, and also the accuracy to predict positive versus negative samples. It was found to be.
So far with these tests, from what we've seen, it's the best test on the market as far as accuracy.
Based on our tests, you're right. We have seen this test to be very, very robust compared to other tests that we have seen.
Okay, so now, can you talk briefly about the interagency process in the United States and where this test is right now with NIH?
Yeah, so this test was submitted to NIH, to actually FDA, for FDA to test itself. This test is currently being tested by NIH, which is collaborating with FDA to test these tests. And this is one of the first tests that was submitted to NIH. We are waiting for the results from FDA. We were told that the test is currently being validated by FDA and NIH. At this time, all we know is that the test is ongoing. We don't have the results for it, but so far we haven't heard anything negative about the test. They have been very positive in our interactions with them.
Right. So I just want to point that out. There's been multiple interactions, and they've been very positive. You just can't release those comments yet.
Yes. I mean, we have sent to FDA all the information. We were one of the first tests to submit to FDA. We have been talking to FDA, over and about with this test.
You mean the first test on this interagency review process? Because there's a couple tests that are reviewed and that are already EUA authorized, but the challenge is that they submitted most of their information before this interagency review even existed.
That is true.
Right. So it's really prestigious if we come out ahead of the interagency review, because they may compare this Reszon test to other tests too. So if this accuracy is even, as close to this at 98 and 100% and 97.9 and 99%, this is really probably the best test on the market.
That is true, but in all fairness, FDA is testing other tests, and FDA's criteria is any test that is 95% and above accurate will be allowed. So even though those tests may not be 99% or 100%, they will be allowed by FDA if they meet the 95% threshold. So, I mean, again, this test comes with a lot of testing. It is being validated very accurately, and it is obviously currently being tested. But we are in the process of working with FDA to figure out what else they may have to ask us.
That's fantastic.
So that's all I had. That's all I had on this test, Vince. I'm happy to answer any other questions you may have.
Well, yeah, we've got a couple questions, and in this case, the questions were submitted already. But one is that this is an EUA authorized, meaning the paperwork is submitted, so you can sell it, correct, and you can use it.
Yes, yes,
Right?
it is permitted by the FDA to sell. It has been filed for EUA. It is compliant with any existing FDA regulations, and it is being legally imported into the country. We have imported several of the thousands of these tests, and we have had no issues with the U.S. government to import these.
And it is being manufactured in Malaysia and not China, and the warehouse is in New Jersey.
That is correct.
Oh, and on this last shipment, we believe it actually came into the country, went through customs in pretty much 7 days, I think, or less.
Yes, yes. I mean, Malaysia is a little better location from shipping point of view. We were able to FedEx it from Malaysia, and it arrived here in about 4 days. And then customs, every one of these tests is tested by FDA before it is released. So we got the test released by customs without any issues.
Yeah, that's an important part, because what's happening with particularly tests coming from China right now, if they're in this country, they're pretty accessible already. But if they're not in this country, there's up to a 2-week hold coming from China if they let it go, and another 2-week hold in the United States plus the shipping time.
That is correct.
Is a problem. And a lot of the EUA approved right now or authorized tests are from China.
That is correct. All of them.
Oh, all of them. Yeah, all of them. So that's something really important. And you expect full EUA authorization on this test shortly, correct?
We do expect that, yes.
All right. Well, I don't know what else to ask other than how did you find this test?
Well, this is a test that came to me for taking to FDA. So I reviewed the information. I wrote the EUA. I'm the one who's talking to FDA on behalf of this company. So it came to me just like any other company where people approach me for helping them with FDA, and that's how I became aware of it.
Are you representing other similar tests?
I have reviewed many tests similar to that. I'm not representing all of them, but I've reviewed several of the tests out there.
Got it. All right. Well, thank you so much. I think this is a pretty clear, easy presentation, and it's going to clear up a lot of misconceptions. Like even me, I thought I knew a little bit more, and I didn't realize that if you have an IgG score, you should probably still get tested. I mean, there's a few things that I picked up here.
Thank you.
I didn't quite realize that you could have IgM and IgG going at the same time either. I figured once you had IgG, you were done with the virus. But then obviously I was wrong, right?
Well, you are not completely wrong. You could be done with the virus and still have IgG, but there is an overlap in the viral infection. Yes, there is. If you were infected a month ago and you are now fully recovered, then most likely you don't have any virus, but you have the IgGs. But if you were recently diagnosed in less than 2 to 3 weeks ago, then it's very possible that the virus is still there. You're still infected.
But what I'm saying is I could have IgM and IgG, and I may not be, I may have the virus, but I might not, correct? So I should get another test.
Yeah, you should always get another test when you get antibody tests.
Right. And then ultimately IgG is good. But at this point,
IgG is good. That basically means you have immunity to the virus now. That is how you measure if somebody is immune to the virus or not.
There are a lot of people that still don't understand what the herd effect is. Could you explain that briefly, then I'll let you go?
Yeah, the herd effect is when too many people, even when above 70% of a population is infected with an infectious agent, what happens is they actually, all these people, they cause a small amount of infection in everybody else. That is 30%, and over time, everybody develops antibodies to that infectious agent. So you no longer see the disease, even though the virus is there. You don't now see symptoms. There are several viruses that we carry in our body. In our evolution over the thousands of years of existence, we have been infected by different pathogens over time, and many of those pathogens are present in our body, but we don't have any adverse effects of those pathogens. That's what we mean by herd immunity. So as a community, even though we may have viruses in our body, we don't have a negative. effect of those viruses. So that's what herd immunity means. Herd immunity, you need to have sufficient number of people infected in a population to develop that kind of overall immunity for everybody. And the number is 70% or above people have to get infected for everybody to become immune to a given virus or any pathogen for that matter.
Got it. So, and the other thing is that at some point here, or maybe right away, those with IgG antibodies, their plasma is probably valuable for future vaccines and whatnot, correct?
It is. It's already being developed. There are many companies that are collecting donated blood from people who were previously infected and using that to purify IgGs to COVID-19, which will be in turn used to treat people who are severely infected with the virus. So that is already an FDA-approved treatment. There are companies who are isolating IgGs from people who were infected and now have recovered.
All right. Well, this is. Thank you so much for your time. This has been most informative.
My pleasure. It has been a pleasure talking to you, Vince.
All right. Thank you. You as well. Always.
Thank you.